Recording, right?
Sir, it's live on Facebook, sir. Sorry, YouTube. It's live on YouTube.
Okay. I think we'll start, right? 3-5 because people are waiting.
Yes, sir,
Huh?
please. Yes, sir.
Okay. Respected Principal Madam, Dr. Anuradha, today's speaker, Dr. Angsuman Bagchi, my dear colleagues and students from various organizations. On behalf of Padma Shri Institute of Management and Sciences, I would like to welcome all participants for the current 5 days program on lecturer series come FDP on protein characterization using bioinformatics tools. I want to mention here learning and progress. These two words become much imperative everywhere in the world. We must keep accelerating our knowledge from time to time. With this objective, our principal is organizing a diverse range of webinar, maybe from functional food to microgreens, from mushroom cultivation to honeybee culture. We also noticed that a dramatic increase in the number of participation. From this, we could conclude people are focusing on learning. And now, learning is not restricted to the answer script of the examination or scoring %. We may not be expertise in all the subjects or all the webinar we participate or all the seminar we participate. But by learning or gaining knowledge, we may utilize somewhere at least for our own work. Time was there, like I will tell about myself. I was thinking bioinformatics, it's a soft core subject. And just to write something in the examination, and that's all. But in my higher study, when we started the experiments, the budget plan directs that we cannot use all the genes or we cannot take all the genes or all the proteins into our study. We need to make it more precise with much accuracy by using in silico tools. Similarly, for statistics, like when we go for publication in a reputed journals, there is a long guidelines which is given by the journals. Even the journal guidelines suggest we cannot use even simple clip art which is there in the internet without the permission. The journal needs a written consent from the copyright holder. Without that, we cannot even use in our publications. So I will tell you one more example. My colleague can make an excellent poster within 5 minutes. Otherwise, we might need the help of a designer. Again, the designer, if he is not from biology background, he will write protons in place of proteins. We faced all this. So time has come to make ourselves skilled with various techniques. Once again, I would like to welcome all the participants of this program who join with interest to learn something new. Without taking much time, now I would like to introduce today's speaker, Dr. Angsuman Bagchi, Associate Professor of Biochemistry and Coordinator for Biochemistry and Coordinator for Bioinformatics Center, Department of Biochemistry and Biophysics, University of Kalyani. Former postdoctoral fellow at Center of Computational Biology and Bioinformatics, Department of Medical and Molecular Genetics, Indiana University, Purdue University, Indianapolis, IUPUI, USA, and Buck Institute, USA. He was recipient of Travel Fellowship Award from National Science Foundation, USA, Visiting Research Scientist Hong Kong Baptist University, HKBU Hong Kong, and INRA France. He has undertaken many research projects and published many research articles in national and international journals to his credit. We are eager to listen to you, Doctor. Over to you, sir.
Thank you very much for the introduction, and I'd like to thank the organizers for inviting me to talk about a little bit of bioinformatics. So I believe that the audience is a mixed bag. That means the audience has some idea about bioinformatics. What I will be talking about is the very basic bioinformatics, that is the different types of databases. And specifically, the databases in biology, in bioinformatics, in the field of bioinformatics, play very crucial roles. And one of the fundamental databases is the NCBI, National Center for Biotechnology Information. So I'm going to share my screen with you. Yeah. So the screen is visible, right?
Yes, sir.
Okay, so it is the Introduction to Biological Databases, NCBI. NCBI stands for National Center for Biotechnology Information, and this database was it was founded in 1988 as a part of the National Library of Medicine at NIH. Initially, the database used to store information regarding the scientific literature. Scientific literature means the different types of journal articles. Also, from then on, the database keeps on collecting information regarding nucleotide and amino acid sequences of proteins and the tools to manipulate them. So this is basically the web interface. So how to access the NCBI? If you look at the web interface, it stores a particular information, all databases. So that all databases gives you an idea about the different types of databases that are tagged to NCBI. There are several other important taggings in the NCBI, that is PubMed, PubMed Central. PubMed and PubMed Central are the literature databases. The bookshelf, it is a very useful one. The bookshelf is the storehouse or the repository of the free ebooks. The BLAST, it is the basic local alignment search tool. It is a tool, it's a software. There are nucleotides and protein information, GEO, PubChem, and various other structure information. Structure means these are the structures of mainly proteins and protein complexes. And PubChem is another database, which is the storehouse, the repository of small molecules. So, the database. So let me give you a brief idea about the biological databases. It is generally considered to be a structured, a specific collection of information. That means structured collection means these are the systematic collection of data. Each data should have a predefined heading. For example, a protein database, as the example is given here, the name of the protein should be there, its length, its amino acid sequences, the percentages of different types of amino acids. All this information should be present in the database. That is why it is called a structured database. That means it should have a specific hierarchy. A perfect database, that means NCBI is an example of a perfect database. It is very easy to search. It is very much annotated. That means annotated means all the information in the NCBI database, in any entry of the NCBI database, is all those entries are defined. There are no entries that are present in any of the NCBI databases that that does not have any kind of definition to it. And it has got various cross references. That means, say you identified a nucleotide sequence from an organism. you deposited your nucleotide sequence in NCBI and published that nucleotide sequence in a journal. So there has to be a linking between the literature and the data that are stored in NCBI. So that is called cross-referencing. And minimum redundancy means repetition. There should be minimum repetition. That means it may so happen that one lab is sequencing, is performing experiments on sequencing of the whole genome of E. coli. Another lab is also doing the same thing. So it may so happen that simultaneously both the labs are trying to deposit their data. So that means there has to be some kind of redundancy because E. coli genome is invariable. It is the same everywhere. So the data should have redundancy. And they NCBI has all these different types of features. So all the types of data that are stored in NCBI, the genome, that is the DNA sequences, the mRNA, the protein sequences, their expression profiles, and the literature information. That means all the central dogma of molecular biology, all those information pertaining to the central dogma of molecular biology, all those information are present in NCBI. So NCBI initially referred to as GenBank. It is a primary sequence database. Now you need to know what a primary sequence database is. A primary sequence database means it is a database which stores the initial data with annotations. Other secondary databases get or acquire information from NCBI and make their own databases. So those are called the secondary databases. So NCBI is, or GenBank is the mother database. And as I told you earlier, that it is linked to many other literature databases like PubMed, PubMed Central, and so on. So mentioned that NCBI is the mother database or the primary database, and the other databases are derived from NCBI. That means there are examples like NCBI protein. Why I am considering the NCBI protein to be a derived database? Because originally what we do is we sequence the nucleotides and store the nucleotide information in NCBI GenBank. Then we extract using the codon table that we extract the amino acid sequences from the nucleotide sequences. So the nucleotide sequences that are present in NCBI serve as the entry point of any analysis. From that entry point, we generate different types of data and store them in other databases like NCBI protein. So those are called the derived databases or the secondary databases. So this kind of a cartoon, that means what is generally being done. That means there are lots and lots of sequencing experiments, and all those sequencing experiments, all those sequences that are coming from those sequencing experiments, because the sequencing experiments have become very routine these days. So all those data that are generated from the various types of sequencing experiments, they are stored in GenBank. And then they are curated. So that is why GenBank is called the mother database or the primary database. Now they are curated by using various computer programs. Those are called the algorithms, and we generate the different types of sequences. One of them is called the RepSeq, the genome assembly, the UniGene, and so on. So this is what I mentioned earlier. Now the GenBank, that is the primary sequence. That this is the as I told you earlier, that GenBank is the basic database of NCBI. So it is the mother database. It stores the most of the information. But the problem with GenBank is that whatever data are being generated by the different types of sequencing experiments, all of them are stored together in GenBank. So there are lots and lots of redundancies. So that is why we need various types of secondary databases to remove those redundancies. So GenBank of NCBI crosstalks with other databases. One of them is DDBJ, database of Japan, and another one is EMBL, European Molecular Biology Laboratory database. So they always there is a crosstalking between all these databases. So this is a traditional GenBank record. So the first line, it gives you an idea about what type of information the database stores. Here it is an mRNA, it's a linear mRNA, and it is a complete CDS. It's accession number. These two are very important. Accession number, version, and GI. Accession number means this is kind of an identifier. So if you know this accession number, you can just type it, and you can you will be directed to this particular database. Otherwise, it will be difficult to search the GenBank. And the version means this point one means initially the data that were deposited in GenBank had this accession number. Then again, the similar experiment was performed on the same data, and a new set of data were generated. The new set of data had some from the previous set of data, and the new set of data is provided with an accession number .1. That means it is a newer version of this, of the originally submitted data. And very stable. That means you can always use this accession number in your journals and papers, manuscripts. This is, as I told you, this keeps on, if you have a first version, then there should be no, no dot. If you have the second version, it should start with .1. If there is a third version, .2, and so on. And this is GI number. This is for the internal use. This is for GenBank identifier. This is for the computer scientists who use this particular GenBank data for his or her own analysis. So we generally, in our manuscript or in our various types of journals, we use this accession number. So this is a closer bird's eye view of a GenBank entry. This is the gene, this is the CDS, and this is the function. So see, these are called the annotations. The annotations. Annotation means these are kind of the definitions, or a kind of evidence, or a kind of explanation to what the possible function or what the possible feature of this entry is. And this is the protein, and this is the amino acid sequence of the protein, which is coming from the nucleotides that are present in this GenBank entry. So this is actually the nucleotide. So this nucleotide information is converted by the codon usage table to the amino acid sequence, and the amino acid sequence is present before. And it also stores how many A's and T's and G's and C's are there in the database. So this is the fundamental of a GenBank entry. This way, we can have a fair amount of idea about how stable and how structured a GenBank entry is. This is the first and foremost important application of NCBI to store the data, to store the genetic information for our analysis. Now, various types of other databases that are also present in NCBI. They are the derived databases, and NCBI stores a number of derived databases, and the data from those derived databases are coming from the original GenBank entry. So this is, as I told you earlier, that this is the CDS translations. That means originally the nucleotide sequence was present, and that nucleotide sequence is translated using the codon usage table, and we are getting the amino acid sequence as mentioned here. This is the uh, identifier. This is the identifier of the amino acid or the protein. Previous identifier, the accession number that I mentioned a few slides back, that accession number is the accession number of the nucleotide entry. This accession number is for the protein. So this is called the RepSeq. RepSeq means this is a reference sequence. This is a derived database. This is coming from NCBI. It stores the curated transcripts and proteins. That means, as I told you earlier, that it contains the different types of data associated with the nucleotide. See, in our biological systems, nucleotides have very limited roles. So the nucleotides are generally converted to proteins by different types of biochemical reactions, you know, the transcription, the translation processes, and the proteins function the majority of the biological reactions. So we need to have an idea about what type of proteins are being developed or are being generated from the nucleotides that are present in the GenBank. So that is the reason we need to have RepSeq. A derived sequence database. Again, I'm saying this is a derived sequence database, or this is a secondary database. Why this is called the secondary database? Because this data is not the original data. We are extracting the data from, or rather I should say, we are extracting the information from the sequence or the nucleotide sequence that is already present in GenBank. So that is why it is called the derived or the secondary database. And it contains different types of information. It contains chromosomal information from human, from microbial, from different types of organelle genomes, and so on. So this is a typical example of the RepSeq accession numbers. It gives you an idea about that. If it is an mRNA, it is represented as NM. If it is a protein, then NP. R. This is non-coding RNA, and so on and so forth. So the RepSeq accession numbers are a little bit different from the accession numbers of GenBank. So far, all good? Everyone is understanding what I'm saying?
Yes, sir. They're all muted, so I don't think they can respond now, sir.
Okay. So if there is any question, you can go ahead and ask me.
At the end, yeah.
Okay. Okay. So this is called GenBank to RepSeq. So that means a GenBank data can be converted to a RepSeq by many different algorithms. This is one of such examples. Now, RepSeq annotations and reagents. This is not so much linked to bioinformatics. This is linked to an experimental like the wet lab biology, that how an mRNA is related and how biochemical experimentations and how the benefits. I told you earlier that the benefits are the non-redundancy. The GenBank and so forth. So that so that
Your
just
voice
by
are
looking
breaking,
at
sir.
the accession number. Yes,
Your voice is breaking, sir.
my voice is breaking.
Yeah. Okay.
Maybe because of some internet. Am I audible?
Yeah, fine now. Yeah.
So as I mentioned earlier, that the accession number, just by looking at the accession number, we can have a direct idea what type of information is there in the database. Sometimes just looking at the GenBank accession number, we cannot ascertain what type of information is there. It may be a nucleic acid, it may be a protein, it may be anything. But the RepSec has that benefit of providing us an idea about the type of information, the category of information that is present there, just by the accession number. As I told you earlier, that NP stands for protein, NR stands for RNA, and so on. So there are other derived databases that are present in NCBI. Those are called the express sequence tags, the DBSNP, the structure, and so on. All these are derived databases. Express sequences means these are that means these are the amino acid sequences which are expressed from a nucleotide. Sometimes a whole genome is not translated. It is always the case. I should not say sometimes, but it is always the case. A few parts of the entire genome, that means which are called the genes, those are translated, and the remaining portions remain untranslated. So the portions, the genes that are translated, so they should create a protein, and the proteins, that means the sequences of the genes are present in the database, a derived database called express sequence database. DB-SNP. SNP means this is single nucleotide polymorphisms. That means small changes in the nucleotide sequences, and those small changes might lead to some kind of aberrations. So those data are present in DB-SNP. The structures of the proteins, those are also present. Those are also very much important, very much vital, and they are present in the different another database which is present in NCBI. And the entire gene information, and so on. So ENTREZ. ENTREZ is a type of retrieval system in NCBI. The ENTREZ, what it does is that ENTREZ helps us to extract the most of the information from a GenBank entry. For example, if we want to extract the abstract of a literature, we should use the ENTREZ algorithm to extract that from PubMed. We can do so for various other databases like the 3D structural database, the nucleic sequences, and so on. So all these, that means the algorithms that we use to collect the features from the different entries in GenBank, and that is called the ENTREZ. Um, this is, I told you, so this is kind of a repetition. So a global query, we can query GenBank in this, like here the query is the Alzheimer's disease, and if we just use this keyword to search GenBank, we get so many different types of data from different types of databases, from PubMed, from books, from genes, from nucleotides, and so on and so forth. This is the ENTREZ system. ENTREZ system uses 30 different types of integrated databases, and the ENTREZ system, the ENTREZ algorithm, can extract the features or information from different 38 and more number of databases that are already present in GenBank. That means, in other words, the ENTREZ algorithm is used to make a correlation or collaborate between the primary GenBank entry and all the other secondary databases that are present in NCBI. So this is kind of, you all know, I believe, that from DNA sequence, we get the nucleotide, and the nucleotide we get the proteins, and from the proteins we can build their structures. So the problem that we face is that the biology is changing every day. Previously, we used to think that the non-coding regions in genomes, they are just junk. They do not have any specificity or any specific functionality. But nowadays, we know there are certain non-coding RNAs, long non-coding RNAs, which are called the LNC RNAs. They take part in many different biochemical processes. So that change in biology, that change in the concept of biology should be reflected in the GenBank databases. So this is the global ENTREZ search, and this is kind of the similar that I showed you earlier. You can type it, you can use any of the keywords here. I am using colon cancer, and we are getting so many different types of data. And you can use the ENTREZ to search a specific, that means search a specific database. Like if we are interested only in the literature database, and we want to know what type of experiments are being done, what type of experiments are being done in colon cancer, in the field of colon cancer, and what type of information are already present in the different types of databases. So in that case, we can use the ENTREZ, and we can use the ENTREZ to search against only the PubMed. So that way we can channelize, our ENTREZ search engine to different types of queries, and uh, it's like the bioinformatics is basically a science that is need based. Like if you are interested only in the genome, then your search, your query should be something. If you are interested in the proteome, your search should be something like this. So in bioinformatics, we cannot say this is a generalized rule to search a database. It is always need-based. So this is an ENTREZ gene record. It looks like any of the GenBank entries, but this is coming from GenBank definitely, but it is a little bit different because the GenBank entry starts with the accession number, but it does not start with the accession number. Rather, it starts with an official symbol. Here, the search is being done with the help of this official symbol, not with the accession number. This is a record, and this is kind of the accession number. This is the chromosomal location, and these are the different types of annotations that can be obtained from this particular gene in ENTREZ. And it is linked to the sequences. These are the sequence, that means these are the accession numbers, the rep sets of the sequences C, N, M, and NP. P stands for, this is protein, this is mRNA. So you can go to these places, and this is of genetic variations. As I told you, this is called the DB SNP, database SNP, single nucleotide polymorphism. genetic variations that are present. Different types of variations. So the most important aspect is that NCBI National Center for Biotechnology Information. NCBI is the mother database. This mother database stores the information. The NCBI GenBank is the primary database. That means whenever there is a sequencing experiment, we store that data in NCBI GenBank. Then, using the ENTREZ algorithm, we can create many different secondary databases. And we store the information, the features that we gather from the nucleotide entry in GenBank into different types of secondary databases. One of the important secondary databases is the RepSec. RepSec stores the nucleotide information, the amino acid information, the RNA information, and so on and so forth. The most important utility of a RepSec data is that a RepSec entry has the accession number, which is very unique. From the accession number, we can identify what type of data are there. For example, if it starts with NP, we can definitely say that, okay, the data is the protein, that means the amino acid sequence, and so on. And the RepSec is a little bit different from GenBank in the sense that GenBank stores all the types of data. That means there might be data redundancies, but RepSec removes the redundant data and stores only those data which are the most relevant ones or the most up-to-date ones. So that is the basic utility of NCBI GenBank and like to update.
Travel.
So any questions from the participants?
Hello. Is it our sir? Okay. Thank you very much, sir, for sharing your knowledge about various database, annotations, accession number, coding DNA sequences, model transcripts, etc. Even any participants would like to ask any questions or clarify any doubt, you can ask.
Sir, the chat box is open. If anybody wishes to pose any questions, they can do so in the chat box, and I shall read out the questions.
Yeah, okay.
Yes, I can see there is one question. Does the accession number and the official symbol has the same function? Yes. Uh, both of them indicate the entry. But the thing is that official symbol is not generally used in the literature. But accession numbers are used in the literature because official symbols, different databases use different types of official symbols to describe the same data. So this is a bug, but accession number is very stable. uniform for all the types of data.
Anyone else? If anyone will raise the hand also, you can unmute so that you can ask directly outside.
Yes, sir. I can do that.
Yeah.
Can there be anomalies with the sequencing? Yes, this is a very vital question. Yes, see, in GenBank, what the customary is, all the labs who are performing sequencing, they get a sequence, they deposit their sequence there. But after that, they can re-verify their sequencing experiments, and there might be errors in the sequencing experiments. So that is why I told you that there are different versions. So the newest version should be considered as the most stable and the most accurate version. So yes, it is true that there might be anomalies. That is why we generally stick to the RepSec, not the GenBank entries always. Runs NCBI, it is NIH, National Institute of Health.
So the next one, how to get accession number. That's what they've asked. The next questions are how to get accession number, or do accession numbers have any validity?
Accession numbers. Accession numbers, this is accession number you cannot get directly until and unless it is written in the manuscript. So accession numbers are generally, we get the accession numbers from literature. And the thing is that it is always the case that it is not always possible to have the accession number of any particular data. So that is why what we do is we use a generic term. For example, in each of the examples, what I showed you is that I use generic terms like colon cancer, Alzheimer's disease. So instead of that, we can directly write that a particular Perkin protein. Perkin protein is the protein which is involved in causing Alzheimer's disease. So we can write directly the Perkin protein, right? So in that case, we get more specific information. But as I told you, that the accession numbers are very difficult to get unless you have the literature for it. So if you have the literature, you can get the accession number from there and you can search the database. But without the literature, it is not possible to get the accession numbers. There is another question.
Sir, it's a follow-up to the earlier question.
We can't rectify any of it at our end, right? So like we will have to go with selecting the different versions. Yes, the thing is that what we do in our bioinformatics experiments is that we take the latest version, because the latest version is considered to be the most stable one. It is the most up-to-date version, and up-to-date versions are considered to be free from errors.
So 2 more questions.
Yes.
Should one refer to the earlier original version or the updated version? Accession number.
Yes.
Should one refer to the original version or the updated version?
The updated one. The original version might contain some errors. Because see, the sequencing experiments are not foolproof.
One more, sir. Just one more.
Without submitting your data to NCBI, um, like, um, like you cannot claim that you are doing the experiment. If you submit your data to NCBI, in that case you can claim that, okay, I did this experiment, so I have the copyright of this experiment of this sequence. Otherwise, someone else might claim that it is his or her copyright, and you won't get any benefit. So that is why what everybody does is they generate the sequence, they submit it to NCBI and get the accession number, and so that they can use the accession number as the lab's copyright.
Sir, one more question, sir. How are redundant sequences identified?
Redundant sequences, because say you are using a generic term, like for example, you are using hemoglobin from human. So you should get uh, a particular sequence, but you generally what happens is you get thousands and thousands of sequences. So those are the redundant sequences. From there, you need to choose the one having the latest version.
I think that is it, sir. That is all the number of questions we have.
Okay, thank you very much, sir, for attending all the queries and sharing your valuable knowledge. Thank you once again, sir. Thank you very much. Announcement for participants. This lecture series will continue for 5 days. Each day we will cover with a different topic, and at the end of the day we will conduct a small test which will be carrying nearly 10 or 15 questions, and participants who score 50% of marks, a certificate will be issued, and feedback form and this for participation to this quiz program will also be shared to the registered email ID. And here I would also like to announce that we will be bringing you a hands-on training program on this bioinformatics, like how to handle the various databases, how to do the data mining, and how to extract or how to identify a particular gene or gene discovery. Subsequently, we will be notifying you to your email ID, or we will communicate to you during the course of time. Thank you very much for providing your valuable time. Thank you, all the participants.
Sir, can I end it?
Yeah, you can end.
Thank you.